Hi everyone, welcome to my channel Pure Biology, and today I come with the video that is the splicing. Okay, splicing is related to the RNA processing, obviously. So first of all, we have to know what is the splicing. The splicing, okay. Before going to this topic, I want to say that if you are the newcomer and this channel is helpful to you, so obviously you can share and subscribe this channel, okay, and press the bell icon for getting all the notifications. Okay, now let's start. So splicing is what? Splicing is the adding of exons and releasing of introns. Now first we have to understand what are called the exons and what are called the introns. Actually the exons, these are called the expressing sequences. Okay, exons are what? Exons are called the expressing sequences and introns are called the intervening sequences. Okay, so actually the genes, you just ignore the sounds that is coming from the outside. Okay, so what are called the exons? Exons are the expressing sequences and introns are called the intervening sequences. So actually the genes have a split structure in which the segments of the coding sequence. So exons are the coding sequences and introns are the intervening sequences. Non-coding sequences. So exons are the coding sequences and introns are the non-coding sequences. And the entire gene is transcribed to yield a long RNA molecule, and introns are then removed by the process splicing. So that means you can. It is a very simple thing that mRNA that contains exons and introns. So exons, that are the coding segment, and the introns, these are the non-coding segments. So we have to when we adjust the mRNA, then in that cases the exons are attached with each other and introns are releasing from the segment. Okay, so here the first type is that the splicing of the GUAG intron. Okay, so here we just remove this line, not types. The splicing of GUAG intron. Okay, so here the first two nucleotides of intron sequence, that are the 5' GU 3', and the last two are 5' AG 3'. Okay, and the pyrimidine region that called pyrimidine tract near 3' end, intron is found in most cases. Where the introns are found, that near the 3' end, and which region? The pyrimidine rich region. Okay, or you can say that the polypyrimidine tract. Next, the process by which or reactions, you can say, that splicing process occurs, or splicing process involved in this reaction. This is called the transesterification reaction, and this transesterification reaction is this contains two phases. One is the first transesterification and then the second transesterification. Okay, and here the splicing of GUAG, that involves two reactions I have already mentioned here. And in the first case, the cleavage at 5' splice site, that occurs by the first transesterification. So in the first reaction or the first transesterification reaction, in that case the cleavage occur at the 5' site. Okay, and this is promoted by hydroxyl group. Where is located this hydroxyl group? This is attached to the 2' carbon of an adenosine nucleotide, and that located within the intron sequences. So here in first case the cleavage of the 5' splice site that occur and promoted by the hydroxyl group at the 2' carbon of an adenosine nucleotide. Clear? And what is the result of that? And the the result of that, this hydroxyl that attack the cleavage, and phosphodiester bond that is broken. Where is the phosphodiester bond remain? That phosphodiester bond remain the 5' splice site and accompanied by the formation of a new 5' 2' phosphodiester bond. So that means this phosphodiester bond that is already present there, that is broken down, and new phosphodiester bond is formed with the 5' and 2' end. Clear? And this is in the next case, the intron has now been looped back on itself to create a lariat structure, and now the introns, they are attached with each other and to form a lariat structure. And the cleavage of the 3' splice site and joining of the exon, it is the next case. So first one is happening what? First the cleavage is seen, phosphodiester bond is broken down, new phosphodiester bond is formed, and that's why the intron, they attach with each other and to form the lariat structure. This is called the first trans-esterification. And in the second esterification, what happened? That is promoted by the obviously 3' hydroxyl group. Where it is attached? It is attached to the end of the upstream exon. Okay, it is attached with the upstream exon, and this group attacks the phosphodiester bond at the 3' splice site. Okay, now this group means what group? This 3' hydroxyl group. This attack the phosphodiester bond where the 3' splice site. Okay, and cleaving that and releasing the intron, and as the lariat structure, and which is converted back to the linear RNA and degraded. And the last step is what? The 3' end of the upstream exon join the newly formed 5' end of the downstream exon and completing the splicing. So second case is what? Very important thing, that hydroxyl group of the group that attached at the end of the upstream exon, it creates the second esterification, and this group attacks the phosphodiester bond where at the 3' splice site, 3' splice site, and cleaving it where the introns are released by forming a lariat structure and degraded. And exons are, in the next case, the exons are at the 5' end and the 3' end, they are attached with each other. And this is the rough diagram. You can see that these are the two exons, and the middle portion, that is the red color marking, this is the intron. Okay, and you can see that this hydroxyl group attached with trying to attach with the GU in the next step. You can see that UG or GU that attached with the A, and here the exon of the 5' 3' that attached with the hydroxyl group. This hydroxyl group create the second esterification with this 3' exon. Clear? And you can see that third stage, the lariat in the form of lariat structure, this AG and AU both are released from this linear mRNA, and here this is, you can see that lariat structure already formed, and the 5' exon and the 3' exon both are attached and to form the linear mRNA. Okay, linear mRNA. Okay, so this is the splicing of AG and GU, I think so. GU and AG. So this is the splicing process of AG and GU, and this is two broad structures. So in the next video I will try to explain about the splicing apparatus. Okay, splicing apparatus. Clear? So already splicing process I have described here. And another splicing process is there, obviously this is called the alternate splicing. So that will another thing, and I will try to describe this alternate splicing in another video. This is the topic where the splicing process I have explained, just GUAG splicing. Clear? Okay, so please continue with my channel for getting the next videos.